Among the vast diversity of life on Earth, the human form presents a striking anomaly. From a zoological perspective, Homo sapiens do not resemble any other primate in a straightforward way, nor do they conform to the broader mammalian pattern. A comparative glance reveals the differences immediately. The human body stands upright on two limbs, balanced with an ease that belies the mechanical difficulty of the task. The skin is mostly bare, with only sparse hair on the head, face, and body, in sharp contrast to the dense pelage of most mammals. The face itself, unusually flat, exposes muscles and features that allow a range of expression rarely matched in the animal kingdom. Eyes are wide, the sclera visible, the lips fleshy and mobile, the nose projecting outward in a manner absent from our closest primate relatives. Such features invite questions of purpose. In evolutionary biology, form is never assumed to be arbitrary. The structural peculiarities of a species are either adaptive, neutral but linked to other changes, or remnants of evolutionary pathways now long abandoned. For humans, the debate has persisted for centuries. Are these traits the functional outcome of natural selection, designed for survival and reproduction in particular environments, or are they accidents of history, products of genetic drift, sexual preference, or constraints imposed by other adaptations? Early naturalists recognized the strangeness of human appearance. In the 18th century, Johann Friedrich Blumenbach, one of the founders of physical anthropology, classified human variation primarily by cranial shape and skin color, but even he noted that the overall morphology of the species seemed uniquely set apart. Charles Darwin, in The Descent of Man, 1871, suggested that many visible human traits could be linked as much to sexual selection as to survival. He observed that elaborate forms in other animals, peacock feathers, deer antlers, or bird songs, often served reproductive display rather than utility. Darwin speculated that hairlessness, facial features, and even the structure of the voice might follow similar logic in humans. Yet functional explanations have also accumulated. The smoothness of human skin, for example, can be framed as an adaptation to endurance running in hot savannas, where sweating provides superior thermoregulation. The upright gait frees the hands for tool use and carrying, while the flat face and reduced jaw structure might reflect dietary changes linked to fire and cooking. Still, none of these theories provides a complete synthesis. Each trait appears to carry multiple possible explanations, and their interconnection suggests a web of evolutionary pressures rather than a single cause. The enigma is compounded by the contrast with our nearest relatives. Chimpanzees, pan troglodytes, and bonobos, pan paniscus, share more than 98% of our DNA sequence, yet they retain long arms suited for arboreal life, projecting jaws with large canine teeth, and thick hair across their bodies. Their eyes reveal little white sclera, and their vocal tracts do not allow the same range of articulated speech sounds. If genetic similarity predicts morphological resemblance, why then do such profound visible differences separate us? Modern paleoanthropology approaches the problem through fossil evidence. Australopithecines, such as Australopithecus afarensis, the species of the famous Lucy skeleton found in 1974 in Hadar, Ethiopia, already exhibited upright walking nearly 3.5 million years ago. Yet their skulls retained jutting jaws, small cranial capacity, and pronounced brow ridges. Over time, through species such as Homo habilis and Homo erectus, we see a gradual flattening of the face, enlargement of the brain case, and modifications to pelvis and limb proportions. These changes appear piecemeal, not synchronized, hinting at complex pressures driving human distinctiveness. Genetic studies deepen the paradox. In 2005, the sequencing of the chimpanzee genome confirmed the minute percentage difference from humans, but also highlighted areas of accelerated change in our lineage, particularly in genes affecting brain development, skin structure, and skeletal growth. The gene FOXP2, identified in the 1990s through studies of a British family with hereditary speech impairment, underwent key mutations in humans that influenced vocal control. The EDAR gene, which affects hair and sweat gland density, shows selective sweeps in certain populations, reflecting environmental adaptation. These molecular insights link appearance not just to environment, but also to the fine-scale workings of developmental biology. Still, the question remains unresolved because human appearance is not simply biological function. It is also profoundly social. The visible face communicates intention, emotion, and identity. Smooth skin becomes a canvas for symbolic modification, through painting, tattooing, or adornment. Hair patterns invite grooming and social bonding. Anthropologist Desmond Morris, in The Naked Ape, 1967, emphasized this duality. The human form serves as both a survival machine and a signaling surface for interspecies interaction. Unlike most mammals, which conceal signals under fur or restrict them to vocalizations, humans externalize identity in their bodies. This duality creates difficulty for explanation. Was hairlessness selected because it aided running under the African sun, or because it displayed health and freedom from parasites to potential mates? Did upright posture evolve for locomotion efficiency, or because it elevated the head and face into a more visible communicative position? Did the flat face emerge solely from mechanical shifts in diet, or because it allowed greater precision in expressing subtle emotions? Each hypothesis carries data in its favor, yet none can exclude the others. The puzzle of human appearance, then, lies not in any single trait, but in the ensemble. No other species combines these particular modifications. The interdependence of skeletal, muscular, neural, and social systems ensures that changes in one domain cascade into others. To understand why humans look so different from every other species, one must trace not isolated features, but the network of transformations that sculpted them. The surface of the human body is a palimpsest of evolutionary pressures, and to decipher it requires following the anatomical and genetic changes step by step, as each adaptation reconfigured the canvas upon which the next would be drawn. It is within this framework that the investigation proceeds, beginning with the most fundamental departure from mammalian norms, the legacy of upright walking, which reshaped the skeleton and set the stage for every other visible distinction that followed. The transformation of the human body into an upright, bipedal form represents one of the most consequential anatomical reconfigurations in mammalian evolution. Quadrupedal locomotion dominates across the class. From rodents to carnivores to ungulates, the distribution of weight across four limbs provides both stability and speed. To rise permanently onto two legs required a wholesale rebalancing of skeletal architecture, musculature, and neural control. The fossil record shows that this was not a sudden shift but a slow, irregular process, unfolding over millions of years, with intermediate stages leaving evidence in the pelvis, femur, and vertebral column. One of the earliest and clearest signatures of habitual bipedalism is preserved in the Litoli footprints, discovered in 1978 by Mary Leakey and her team in northern Tanzania. These impressions, dated to approximately 3.6 million years ago, were left by hominins walking across volcanic ash. The prints reveal a well-developed arch, a forward-pointing big toe, and a heel-to-toe gait remarkably similar to that of modern humans. Such evidence indicates not a tentative experiment, but an already functional adaptation. The individuals who left those traces walked with a stride balanced for distance. The skeletal adjustments required for this posture are extensive. The pelvis narrowed and rotated, becoming bowl-shaped to support the abdominal organs against gravity. This change altered the birthing canal, a modification that would later intersect with the evolution of larger cranial capacity. The femur angled inward from hip to knee, bringing the knees beneath the center of mass and permitting balance during single-leg stance. The spinal column developed pronounced lumbar curvature, forming an S-shaped profile that redistributed weight over the pelvis rather than forward of it. Without such adaptations, upright walking would collapse under its own instability. Yet these gains came with costs. Comparative anatomy shows that the lower back of humans bears stresses unknown to quadrupeds. Intervertebral discs in the lumbar region are compressed by vertical load, leading to susceptibility to herniation and chronic pain. The hip joint, remodeled for upright stance, is vulnerable to degenerative arthritis, particularly in populations with extended longevity. Knees, tasked with absorbing shock across millions of cycles, often develop wear on articular cartilage, a pathology uncommon in other primates. Evolution did not design a flawless mechanism. It reshaped an existing quadrupedal template into a compromise between efficiency and fragility. The efficiency itself lies in energy conservation. In the 1980s, anthropologists Peter Rodman and Henry McHenry compared the metabolic costs of bipedal and quadrupedal locomotion in primates. Their findings indicated that bipedalism consumes less energy per unit distance at moderate speeds, especially when traveling long ranges. In the open landscapes of East Africa, where early hominins foraged and scavenged, this advantage would have mattered greatly. Covering distances with reduced caloric expenditure allowed broader ranges of movement and access to dispersed resources. Endurance running studies, such as those conducted by Daniel Lieberman and Dennis Bramble at Harvard University, later expanded on this idea, proposing that humans evolved not merely to walk but to run for extended periods, aided by sweat cooling and long tendons acting as elastic springs. The arms, released from locomotor duties, followed a separate trajectory. Reduced in length relative to legs, they no longer required adaptations for brachiation or knuckle walking. Instead, their mobility and dexterity expanded, permitting the manipulation of tools, the carrying of offspring, and the transport of food. Shoulder anatomy retained considerable flexibility, a remnant of arboreal ancestry, but its integration into an upright frame created new functional possibilities. This shift in limb proportion produced a distinctive silhouette, unlike any other primate, long lower limbs stabilizing the body in stride, with shorter upper limbs capable of fine manipulation. Balance was mediated not only by bone and muscle, but also by neural recalibration. The vestibular system of the inner ear, responsible for detecting motion and orientation, became tuned to stabilize gaze during upright movement. The cerebellum, coordinating posture and locomotion, expanded its circuitry to integrate sensory inputs required for balance on 2 feet. Studies using motion capture analysis of walking gaits demonstrate the subtle complexity of this coordination, involving anticipatory adjustments to maintain equilibrium during each step. The neural demands of bipedalism may have indirectly encouraged the growth of cognitive complexity, as locomotor control intertwined with planning and foresight. The vulnerability introduced by upright walking was significant. A narrow pelvis, while efficient for locomotion, created a tight channel for childbirth. Obstetric complications are virtually unknown in other primates, whose wider pelvic structures accommodate neonates more easily. In humans, the head of the newborn only just passes through the birth canal, often requiring rotation during delivery. Anthropologist Sherwood Washburn termed this the obstetrical dilemma, a trade-off between efficient bipedalism and increasing brain size. This constraint forced humans into a pattern of giving birth to highly altricial infants, dependent and immature, a factor that shaped social organization and parental care. Fossilized skeletons illustrate the gradual resolution of these tensions. The pelvis of Australopithecus afarensis is narrower than that of apes, but broader than that of later Homo. Femoral fragments from Homo erectus, dated to 1.6 million years ago, reveal limb proportions already close to modern humans, indicating adaptation to long-distance travel. The Turkana boy skeleton, discovered by Kamoya Kimyu in 1984 on the shores of Lake Turkana, Kenya, showed limb lengths nearly indistinguishable from those of modern adolescents, demonstrating that the transition had reached near completion by that time. Archaeological traces of hunting and scavenging activities suggest that these locomotor changes were not merely incidental. Stone tools associated with Homo habilis and later species indicate transport of raw materials over distances, an activity dependent on freed hands. Cut marks on animal bones show systematic butchery, suggesting cooperative foraging that required carrying meat back to central locations. The body, configured for walking upright, became the vehicle for these behaviors. The outward form that strikes the eye as so different was a reflection of profound ecological strategy. Yet each structural gain generated new pressures. A body balanced vertically is inherently less stable than one supported on four limbs. Falls, fractures, and injuries posed persistent risks, as skeletal remains frequently a test. Anthropological analyses of early hominin fossils, such as the partial skeleton of Australopithecus sediba discovered in South Africa in 2008, show trauma patterns consistent with falls from height, possibly reflecting both arboreal activity and difficulties with full terrestrial balance. The posture that granted energy efficiency also invited instability. The human figure, with its narrow pelvis, elongated lower limbs, and upright axis, remains an experiment still burdened by compromises. Its very efficiency is contingent on conditions that can fail under strain, age, or disease. The outward image of smooth, upright poise conceals an architecture of constant tension, every step a negotiation between gravity, balance, and momentum. From this architecture emerges a body set apart from all others, and it is within this altered frame that further transformations of the face, the skull, and the communicative surfaces of the body began to unfold. When the human figure is examined, one of the most striking distinctions lies not in posture or limb proportions, but in the face. The human face presents a configuration unusual among mammals: a flattened midline profile, reduced jaws, a protruding nasal bridge, fleshy lips, and eyes with large visible sclera. These features combine to form a communicative surface unlike that of any other primate. While locomotion altered the skeleton, the face became the canvas upon which both biological necessity and social interaction were inscribed. Comparative anatomy demonstrates the divergence clearly. In chimpanzees, gorillas, and orangutans, the prognathous snout projects well beyond the plane of the eyes, housing large canines and extensive masticatory musculature. The brow ridges are pronounced, the nose broad and flat, the lips thin. In humans, the maxilla and mandible recede beneath the cranium, the teeth are smaller, and the chewing muscles less massive. This flattening of the profile, known as orthognathism, reshapes not only the mechanics of eating but also the visibility of facial musculature. Subtle contractions around the eyes and mouth become readable at a distance, allowing expressions to serve as rapid, nuanced signals. The reduction of jaw size has long been linked to dietary changes. Richard Wrangham and colleagues have argued that the control of fire and the cooking of food, a practice at least 400,000 years old and possibly much older, decreased the requirement for large teeth and powerful chewing muscles. Softer diets reduced selective pressure on massive jaws, permitting cranial reorganization. The temporalis muscle, which in apes extends high along the skull, shrank in humans, freeing space for cranial expansion. The result was a face less dominated by bone and more by the play of expression. Paleoanthropological evidence supports the timing of this transformation. Fossils of Homo erectus show smaller molars and thinner enamel than those of Australopithecines, alongside reduced prognathism. Later species, including Homo heidelbergensis and Homo neanderthalensis, display continued reduction, though Neanderthals retained large nasal apertures and heavy brow ridges. Modern Homo sapiens, appearing around 300,000 years ago, exhibit the most reduced and delicate facial form. The trajectory suggests a cumulative process, with mechanical, dietary, and social factors reinforcing one another. Among the most distinctive features is the nose. The projecting nasal bridge and narrow nostrils are rare in mammals, where external noses are typically short and broad. Physiological studies indicate that the human nose functions as an air conditioner, warming and humidifying inhaled air in cold, dry climates and filtering particles before they reach the lungs. Variability in nasal shape across populations corresponds to climatic adaptation. A 2017 study led by Arslan Zaidi analyzed nasal width in relation to humidity and temperature, finding statistically significant correlations between narrower noses in cold, dry regions and broader noses in warm, humid zones. Yet beyond physiology, the nose shapes the visual identity of the face, creating a midline prominence that transforms how features are perceived. Equally striking is the eye. In most primates, the sclera, the white outer layer of the eyeball, is pigmented dark, minimizing contrast with the iris and reducing visibility of gaze direction. In humans, the sclera is depigmented and conspicuously large. This creates high contrast with the iris and pupil, making eye movements easily visible to others. Evolutionary psychologists Michael Tomasello and Josep Coll have argued that this adaptation facilitates cooperative communication, allowing humans to follow one another's gaze with precision. Infants as young as 6 months track eye direction more effectively in humans than in apes, evidence that the visible sclera underpins social cognition from an early age. Facial musculature completes this system. Humans possess approximately 43 individual facial muscles, with an exceptional degree of fine motor control. Studies by Paul Ekman and Wallace Friesen in the 1970s catalogued these movements in the facial action coding system, demonstrating how combinations of contractions produce universally recognizable expressions of emotion such as fear, anger, happiness, sadness, and surprise. Unlike vocalizations, which can be masked or withheld, facial expressions occur rapidly and often involuntarily, making them reliable indicators of internal state. The flattened human face exposes these movements to view, transforming biology into a communicative medium. Teeth, though reduced, also contribute to expression. The human smile, exposing small, evenly spaced teeth framed by mobile lips, is unlike the tooth-bearing threat displays of primates. Ethological studies show that smiling functions as an affiliative signal, disarming aggression and inviting cooperation. Over evolutionary time, what began as a submissive gesture may have been reconfigured into a sign of social bonding. The delicate form of the jaw, combined with lips capable of complex shaping, enables this transformation of function. The interplay between structure and expression becomes evident when considering sexual selection. Traits that signal health, youth, and symmetry are often magnified in human preferences. Facial symmetry, measurable by comparing left-right ratios, is consistently rated as attractive across cultures, possibly because it reflects developmental stability. Full lips, clear skin, and proportionate features are likewise associated with reproductive fitness. The human face, more than any other anatomical feature, became a site where natural selection and sexual selection converged, each shaping its contours in overlapping but distinct ways. Cross-cultural research underscores the universality of these signals. Anthropologist David Buss's surveys across 37 societies in the 1980s revealed consistent valuation of facial attractiveness, despite wide differences in environment and tradition. Preferences may vary in detail, such as the relative weight given to facial roundness or angularity, but the centrality of the face as a measure of desirability remains constant. This universality suggests that the communicative power of the face is deeply rooted in human biology, not merely cultural construction. Yet the face is not static. Developmental biology shows that infants possess rounder faces, larger eyes relative to head size, and smaller jaws. These neotenous features elicit caregiving responses from adults, an effect Konrad Lorenz termed the Kindchenschema. As humans age, the jaw elongates, brow ridges develop, and facial fat distribution changes. The shifting form across the lifespan allows the face to signal age, maturity, and social role, extending its communicative range beyond immediate expression. Still unresolved are the precise evolutionary pressures that privileged such expressiveness. Some argue that cooperative hunting and group living demanded transparent communication of intention. Others emphasize the role of mate choice, where faces served as primary arenas for selection. A third perspective points to the interaction of dietary mechanics, brain expansion, and speech, each constraining and reshaping the bones of the face. The debate continues, not over whether the human face is unique, but over which forces weighed most heavily in sculpting it. The distinctive arrangement of eyes, nose, lips, and jaw is more than ornament. It is a functional system integrating physiology, communication, and social dynamics. Yet even as the face became the most visible site of human distinctiveness, another transformation unfolded across the body, stripping away a feature that unites nearly all mammals and exposing the skin as a new and unprotected surface. Among the mammals, the absence of a dense coat of body hair in humans remains one of the most conspicuous anomalies. Fur serves multiple critical functions across species. It insulates against cold, shields against ultraviolet radiation, and protects the skin from abrasions and parasites. Its loss in humans, leaving a largely exposed surface interrupted only by localized patches of hair, requires explanation. The evolutionary pressures that stripped the body bare have been debated for more than a century, with competing hypotheses invoking climate, disease, social signaling, and sexual preference. Physiological data provide a starting point. The human integumentary system contains between 2 and 5 million sweat glands, of which approximately 2 to 3 million are eccrine glands distributed across nearly the entire body. These glands secrete a watery fluid that evaporates to dissipate heat. In contrast, most mammals rely on panting or localized sweating through apocrine glands. The extensive human eccrine network, unmatched in density among mammals, permits continuous cooling during sustained exertion. Without the barrier of a thick fur coat, evaporative efficiency increases dramatically. Laboratory experiments conducted by Peter Wheeler in the 1990s quantified heat loss in simulated savanna conditions, demonstrating that a naked skin surface exposed to moving air could sustain endurance activities that would overheat a furred mammal. The loss of fur, therefore, is closely tied to thermoregulation. This physiological adaptation gains meaning when paired with ecological context. Endurance running, proposed by Daniel Lieberman and Dennis Bramble as a central human adaptation, depends not only on musculoskeletal form but also on temperature management. Pursuit hunting, documented ethnographically among the San of the Kalahari and the Tarahumara of northern Mexico, involves tracking animals across long distances until prey collapses from hypothermia. Such hunting methods would be impossible without the dual system of bipedal efficiency and naked skin sweating. Fur loss, in this view, was less a vulnerability than an asset in hot, open environments. Yet climate alone does not resolve the question. If fur loss was solely adaptive for heat dissipation, one might expect similar changes in other large-bodied mammals of the savanna. Zebras, wildebeest, and antelope retained coats while evolving behavioral strategies for heat avoidance. The peculiarity of human nakedness has led to alternative theories. One influential proposal, advanced by Mark Pagel and Walter Bodmer, suggests that reduced hair offered protection against ectoparasites such as lice, ticks, and fleas. Bare skin allows easier detection and removal of parasites, reducing disease transmission in group-living species. This perspective finds support in genetic evidence. Studies of louse divergence indicate that head lice and body lice split into distinct lineages around 170,000 years ago, coinciding with the adoption of clothing, which created a new niche for parasites once the protective fur coat had disappeared. Sexual selection has been invoked as another force. Darwin himself speculated that smooth skin may have become aesthetically desirable, with individuals preferring less hirsute partners. Over generations, such preferences could amplify hair reduction beyond its functional benefits. This hypothesis gains plausibility from the fact that body hair distribution varies by sex and age, with males typically exhibiting denser terminal hair on the chest, face, and limbs. These sexually dimorphic patterns suggest that hair serves a role in signaling maturity and secondary sexual traits, even as the general covering was lost. The persistence of scalp hair complicates the picture. Unlike body hair, which is fine and sparse, scalp hair grows continuously and densely. Its length and variability across populations point to a distinctive evolutionary trajectory. One functional explanation emphasizes protection against solar radiation. Long hair shades the head, while tightly curled hair in equatorial populations creates an insulating layer that limits heat gain. Another perspective views scalp hair as a sexually selected ornament, its growth and color variation serving as indicators of health and vitality. Archaeological evidence of hair grooming tools, such as bone combs found at Upper Paleolithic sites, attests to the antiquity of deliberate hair management, reinforcing the role of hair in social and aesthetic domains. The biochemical substrate of skin, melanin, also interacts with hairlessness. With fur gone, skin became the primary defense against ultraviolet radiation. Melanocytes in the epidermis produce eumelanin and pheomelanin, pigments that absorb UV rays and prevent DNA damage. Genetic studies identify variants in genes such as MC1R, SLC24A5, and KITLG that influence melanin production and distribution, showing evidence of strong selection across populations. The interplay between naked skin and pigmentation indicates a dual adaptation. Exposure necessitated a chemical shield, while pigmentation itself shaped the visible spectrum of human diversity. Beyond biology, the disappearance of fur redefined social interaction. Bare skin became a surface for touch, grooming, and visual display. Primate studies show that grooming is central to social cohesion. In humans, the exposed surface expanded the possibilities of contact, transforming tactile interaction into a primary mode of bonding. The lack of fur also created a new canvas for cultural practices. Painting, tattooing, and ornamentation became feasible in ways unavailable to fur-covered species. Archaeological discoveries of ochre use, such as those at Blombos Cave in South Africa, dating to 75,000 years ago, suggest that pigmentation of the skin may have carried symbolic meaning, embedding the naked body in systems of identity and ritual. The debate over the precise trigger of fur loss remains unresolved. Climate, parasites, sexual selection, and cultural reinforcement all present plausible mechanisms, and their interaction may have been synergistic. What is clear is that the disappearance of fur did not simply expose the body, but reconfigured the relationship between physiology, ecology, and society. Naked skin altered the dynamics of heat exchange, disease vulnerability, and aesthetic preference, while also opening the body to symbolic modification and social signaling. The transformation was not without risks. Exposed skin became vulnerable to injury, requiring rapid healing mechanisms and robust immune responses. It also heightened reliance on pigmentation, shelter, and clothing. The balance between vulnerability and opportunity shaped the trajectory of human distinctiveness, for the stripped surface both revealed and demanded further adaptation. With a canvas of skin exposed to light and climate, variation in pigmentation began to emerge as one of the most visible axes of human diversity, a change that carried both biological necessity and social consequence. The exposure of human skin to direct sunlight created an evolutionary problem that demanded biochemical solutions. Unlike furred mammals, which shield ultraviolet radiation with dense hair, humans rely on pigmentation as their primary line of defense. Melanin, produced in specialized cells of the epidermis known as melanocytes, absorbs and scatters ultraviolet, UV, photons, reducing DNA damage and protecting folate, a vitamin critical for embryonic development and cellular repair. The distribution, density, and type of melanin across human populations represent one of the most visible markers of adaptation to local environments, reflecting a delicate balance between the destructive and beneficial effects of solar radiation. The physiology of melanin production is well characterized. Within melanocytes, the enzyme tyrosinase catalyzes the conversion of the amino acid tyrosine into dopaquinone, which then follows biochemical pathways yielding either eumelanin, a dark brown-black pigment, or pheomelanin, a lighter red-yellow pigment. The relative ratio of these two pigments, combined with the number and size of melanosomes in keratinocytes, determines skin tone. Darker skin results from larger melanosomes filled with eumelanin, dispersed individually throughout the cell, whereas lighter skin involves smaller melanosomes that cluster together. Genetic studies have revealed the specific loci under strong selection. Variants in MC1R regulate the switch between eumelanin and pheomelanin. In African populations, MC1R is highly conserved, indicating strong pressure to maintain eumelanin-rich pigmentation for UV protection. In contrast, European populations show greater polymorphism, including alleles that produce red hair and light skin, reflecting relaxation of selection in low UV regions. Genes such as SLC24A5 and SLC45A2, both influencing melanosome function, underwent rapid selection in Eurasian populations, with derived alleles reaching frequencies above 90% in Europe within the last 20,000 years. These mutations correlate with lighter skin, an adaptation believed to facilitate vitamin D synthesis under limited solar exposure. The dual role of UV radiation complicates the picture. On one hand, excessive UVB exposure damages DNA, generates free radicals, and depletes folate, leading to neural tube defects in embryos and reduced fertility in men. On the other hand, moderate UVB stimulates the conversion of 7-dehydrocholesterol in the skin into pre-vitamin D3, which is later hydroxylated in the liver and kidneys to form calcitriol, essential for calcium absorption and bone health. Natural selection therefore operated on a narrow range. Dark pigmentation provided necessary protection in equatorial zones, while lighter pigmentation offered a survival advantage in northern latitudes where dietary sources of vitamin D were limited. Anthropological evidence supports this model. Nina Jablonski and George Chaplin, in a landmark 2000 study, mapped global skin color distribution against UV radiation intensity, finding a striking correlation between darker pigmentation near the equator and lighter pigmentation at higher latitudes. Exceptions exist, notably among the Inuit of the Arctic, whose relatively dark skin is explained by a diet rich in vitamin D from marine sources, reducing the need for lighter pigmentation. Such cases illustrate the interaction between environment, diet, and genetics in shaping human appearance. Pigmentation also intersects with disease resistance. Dark skin reduces the risk of skin cancers such as melanoma, although these diseases generally affect individuals after reproductive age, limiting their impact on selection. More significant is the role of melanin in protecting folate, since folate deficiency directly impairs reproductive success. Conversely, lighter skin in northern latitudes mitigated rickets, a bone-deforming disease caused by vitamin D deficiency, which was particularly severe in urban populations during the Industrial Revolution, where smog reduced sunlight exposure. Historical records from 19th century Britain document widespread childhood rickets, highlighting the ongoing interaction between environment and pigmentation. Variation within populations reveals additional complexity. Sexual dimorphism in skin color is well documented. On average, women exhibit lighter skin than men across diverse populations. This difference is thought to reflect reproductive physiology, as lighter skin facilitates greater vitamin D synthesis during pregnancy and lactation, when calcium demands increase. Studies in the mid-20th century by anthropologist John Rillethford confirmed this pattern across multiple continents, suggesting that hormonal regulation of pigmentation amplifies the evolutionary trade-offs between protection and synthesis. Cultural practices have long interacted with biology. Archaeological evidence from ancient Egypt shows the use of ochre and other pigments applied to skin, possibly as both sunscreen and cosmetic enhancement. Modern sunscreen technologies, though chemically distinct, represent continuity in the effort to mediate between solar exposure and skin vulnerability. Clothing, too, altered selective pressures. Genetic estimates place the origin of body lice around 170,000 years ago, coinciding with widespread use of clothing, which reduced dependence on skin pigmentation alone as a barrier against UV. The invention of shelter and clothing shifted the intensity of selection, but did not erase the fundamental role of melanin. The social meanings attached to pigmentation complicate its biological story. Across history, skin color has been laden with symbolic, aesthetic, and hierarchical associations, often detached from its environmental basis. In some cultures, lighter skin became linked with social class, as in ancient Rome and Han China, where elites avoided outdoor labor. In others, darker skin has been valorized, as among pastoralist groups in Africa, who equated deep pigmentation with health and vitality. These cultural overlays do not alter the biological origins of pigmentation, but demonstrate how human distinctiveness extends beyond adaptation into constructed systems of meaning. Scientific research continues to refine the chronology of pigmentation evolution. Ancient DNA studies, such as those analyzing Mesolithic hunter-gatherer remains from Labrana, Spain, dated to 7,000 years ago, reveal individuals with dark skin pigmentation alleles combined with light eye color variants, combinations not commonly seen in modern populations. Such findings indicate that the current distribution of pigmentation traits is relatively recent, shaped by migrations, interbreeding, and agricultural shifts. The story of human skin color is not static but dynamic, with rapid change occurring within the last 10 millennia. The exposed surface of the human body carries both the imprint of solar radiation and the record of adaptation. The spectrum of pigmentation across humanity reflects a continuous negotiation between protection, synthesis, and environment, mediated by genes under intense selection. Yet skin color is only one aspect of the broader transformation of the head and body, for beneath the surface, the architecture of the skull itself underwent expansion and reshaping, creating a cranial vault of unprecedented size and form. Among the anatomical transformations that set humans apart, none is more conspicuous than the size and shape of the cranial vault. Relative to body mass, the human brain is exceptionally large, averaging around 1,350 cubic centimeters in volume, nearly 3 times that of chimpanzees. The expansion did not occur uniformly. It reshaped the skull, displaced facial structures, and imposed mechanical constraints that reverberated through the entire body. The form of the human head is not simply the container of cognition, but a visible trace of evolutionary compromises between brain growth, locomotion, reproduction, and communication. Fossil evidence reveals the tempo of enlargement. Early Australopithecines, dating between 4 and 2 million years ago, possessed cranial capacities between 400 and 500 cubic centimeters, only slightly above apes. With Homo habilis, emerging around 2.4 million years ago, volumes increased to roughly 600 to 700 cubic centimeters. Homo erectus, widespread by 1.8 million years ago, reached 800 to 1,100 cubic centimeters, approaching the lower range of modern humans. By the time of Homo heidelbergensis and Neanderthals, volumes exceeded 1,200 cubic centimeters, in some cases surpassing modern averages. The fossil sequence demonstrates not a steady climb but episodes of rapid expansion, likely linked to ecological and social thresholds demanding higher cognitive capacity. The mechanics of cranial expansion are evident in the bones themselves. In apes, the skull is dominated by the face, with a receding forehead and protruding muzzle. In humans, the frontal bone extends vertically, forming a high forehead, while the parietal bones arch outward to accommodate expanded cerebral hemispheres. The occipital bone shifts under the skull, bringing the foramen magnum into alignment with upright posture. These changes create a rounded vault, reducing heavy brow ridges and projecting jaws in favor of a bulbous cranial dome. Developmental biology explains how such form is achieved. Human infants are born with unfused cranial sutures and fontanelles, gaps of flexible connective tissue that permit the skull to deform during birth and expand during postnatal brain growth. Neural tissue grows rapidly in the first 2 years, doubling in size, and the sutures remain open into adolescence to accommodate further development. This prolonged plasticity contrasts with apes, where sutures close earlier and brain growth decelerates more quickly. The extended period of cranial expansion reflects the protracted trajectory of human development, binding skull shape to life history strategy. The mechanical consequences of cranial enlargement are severe. The obstetric dilemma, first articulated by Sherwood Washburn in the 1960s, highlights the conflict between a narrow bipedal pelvis and the large fetal head. Human childbirth is uniquely difficult among primates, often requiring rotational maneuvers of the infant within the birth canal. High maternal mortality rates in pre-modern populations testify to the risks imposed by this constraint. Anthropologist Karen Rosenberg has documented the evolutionary solutions: earlier birth of neurologically immature infants, cooperative birthing practices, and possible pelvic variation among populations. The distinctive shape of the human skull cannot be separated from the social adaptations it compelled. The surface anatomy of the skull further records the interplay between brain and face. Reduction of the masticatory apparatus, noted in earlier transformations, coincided with an expansion of the frontal and temporal lobes. The zygomatic arches, massive in Australopithecines to house chewing muscles, diminished, freeing lateral cranial space for neural tissue. The supraorbital torus, prominent in Neanderthals, reduced in modern humans, allowing thinner bone between the brain and facial skin. These structural refinements enhanced the expressiveness of the face, but also reflected underlying neurological reorganization. Endocasts, the internal impressions of fossil crania, provide evidence of cortical shifts. Ralph Holloway's comparative analyses in the late 20th century showed that early Homo species already displayed reorganization of Broca's area, a region associated with speech production. Enlargement of the parietal lobes in later humans correlates with visuospatial integration, tool use, and symbolic capacity. The globular shape of modern human crania, distinct from the elongated forms of Neanderthals, appears linked to expansion of the cerebellum and parietal association cortices. Genetic evidence supports this interpretation. Mutations in genes such as RGAP11B and SRGAP2C, unique to humans, promote neural progenitor proliferation, increasing cortical surface area. The outward appearance of the skull reflects these interior transformations. A vertical forehead signals expansion of the prefrontal cortex, associated with planning and decision-making. Rounded parietals mark increased integration of sensory and motor functions. The diminished muzzle and retracted face expose the cranial vault, making the head appear disproportionately large relative to the body, especially in infancy. This disproportion has aesthetic and social consequences. Large-headed infants elicit caregiving responses, a phenomenon rooted in the perceptual bias toward neotenous traits. Cultural practices further accentuate cranial distinctiveness. Artificial cranial deformation, documented archaeologically in the Andes, Central Asia, and among early European groups, involved binding infants' heads to alter skull shape. Such modifications did not increase brain size but exaggerated vault forms, demonstrating awareness of the head as a locus of identity and status. The skull became not only a product of evolutionary pressures, but a surface for deliberate reshaping, embedding biology within culture. Yet the enlarged cranial case carries vulnerabilities. The metabolic demands of the brain are enormous. Approximately 20% of human resting energy expenditure sustains neural tissue, despite the brain comprising only 2% of body mass. This requirement imposes dietary pressures, favoring energy-dense foods and possibly driving the development of cooperative hunting, cooking, and food-sharing practices. Failure to meet these demands results in developmental deficits, making the cranial vault not merely an anatomical feature, but a constant reminder of nutritional dependency. The skull's form also heightened risks of trauma. Thin cranial bones, necessary for expansion, are more susceptible to fracture. Paleopathological studies reveal healed cranial injuries in Neolithic skeletons, evidence of both interpersonal violence and survival after trauma. The brain's protection demanded social mechanisms of care and recovery, reinforcing the interdependence that marked human communities. The skull became not only the housing of cognition, but also the most fragile and guarded site of the body. The visible differences between human and ape heads, so easily recognized in museum displays, stem from layers of compromise: pelvic constraint, metabolic cost, developmental timing, and social reinforcement. Each left its trace in the contours of the cranial vault and the proportions of the face. But the skull was not the only structure reshaped by the requirements of communication and cognition. Beneath the cranial base, the descent of the larynx and the restructuring of the vocal tract opened a new dimension of human distinctiveness, one that transformed anatomy into the possibility of language. The transformation of the human throat and vocal tract created an anatomical system capable of producing the full spectrum of spoken language. This modification is striking in evolutionary terms. No other primate possesses the same capacity for controlled, complex vocalization. The descent of the larynx, coupled with reshaping of the pharyngeal cavity and fine motor control of respiration, enabled humans to generate sounds with unprecedented variability. Yet this adaptation was not without risks, as the reconfiguration of the airway introduced vulnerabilities absent in other mammals. The larynx, or voice box, houses the vocal folds, which vibrate as air passes from the lungs to create sound. In most mammals, including primates, the larynx is positioned high in the throat, allowing the epiglottis to overlap the soft palate. This arrangement permits simultaneous breathing and swallowing, reducing the risk of aspiration. In humans, however, the larynx descends during infancy, enlarging the supralaryngeal vocal tract. This extended resonating chamber, shaped by the tongue, lips, and soft palate, enables the articulation of distinct vowels and consonants. Ontogeny mirrors this evolutionary change. Newborn humans, like other mammals, have a high larynx, but between 3 months and 2 years, it begins to descend. By adolescence, particularly in males where hormonal influences extend the vocal folds, the larynx reaches its mature position. This descent introduces a permanent risk of choking, as food and liquid can enter the trachea. Mortality statistics confirm the cost. Accidental airway obstruction is far more common in humans than in other primates. The persistence of this arrangement indicates that the communicative benefits outweighed the dangers. Acoustic studies highlight the uniqueness of the human tract. Peter Ladefoged's phonetic research demonstrated that the range of vowel space, defined by the first and second formant frequencies, is far greater in humans than in chimpanzees or macaques. While primates produce graded calls and screams, their vocal tract configurations do not allow the discrete, contrastive vowel categories required for language. Human speech, by contrast, can exploit the full potential of a two-tube system, an oral cavity shaped by the tongue and a pharyngeal cavity extended by laryngeal descent. The neural basis of vocal control is equally distinctive. The motor cortex of humans contains direct corticobulbar projections to the nucleus ambiguus, which innervates laryngeal muscles. This pathway, rare in other primates, grants fine voluntary control over phonation. Coupled with specialized regions such as Broca's area in the inferior frontal gyrus and Wernicke's area in the superior temporal gyrus, this circuitry underlies the coordination of sound, syntax, and meaning. Comparative neuroanatomy confirms that while apes possess homologous regions, they lack the dense connectivity required for fluent speech. Fossil evidence of vocal capability is indirect but informative. The discovery of a modern-like hyoid bone in Neanderthals at Kebra Cave, Israel, dating to 60,000 years ago, suggests that they shared aspects of the human vocal tract. Endocasts of hominin skulls indicate expansion of Broca's area by Homo erectus. Genetic studies add further support. The FOXP2 gene, crucial for speech motor control, contains human-specific mutations absent in chimpanzees but present in Neanderthals, implying that elements of linguistic capacity predate modern humans. The exact phonetic range of archaic humans remains debated, but evidence suggests that the anatomical prerequisites for speech evolved gradually rather than suddenly. The human voice, once established, became more than a mechanical signal. Acoustic variation carries information about age, sex, and health. Male voices deepen at puberty due to androgen-driven lengthening of the vocal folds, producing a lower fundamental frequency. Cross-cultural studies show that deeper male voices are often rated as more dominant, while higher-pitched female voices are associated with youth and fertility. These associations link voice to sexual selection, embedding acoustic traits within the broader matrix of human appearance. The expressive range of the voice extends beyond speech. Singing, chanting, and rhythmic vocalization are documented in all known cultures. Ethnomusicological research suggests that collective vocal expression strengthens group cohesion, synchronizing emotional states through shared rhythm and harmony. From an evolutionary perspective, such practices may have enhanced cooperation, reducing intragroup conflict. The physical modifications of the throat enabled not only linguistic exchange, but also ritualized sound that reinforced social bonds. Yet the vulnerability introduced by these modifications remains pronounced. The shared passage for food and air means that even minor coordination errors can lead to fatal aspiration. The prominence of choking hazards in archaeological records is difficult to establish, but modern medical statistics reveal the magnitude of the risk. The fragility of the arrangement illustrates the evolutionary trade-off: communicative power purchased at the cost of safety. The throat also interacts with facial expression and breath control. Speech requires not only phonation but precise modulation of airflow, coordinated with lips, teeth, and tongue. This integration made the human face a dual instrument, expressive through muscle contraction and articulate through vocal shaping. The visibility of the mouth during speech reinforced the communicative system, as observers could both hear and see cues of intention. This multimodal channel magnified the effectiveness of social interaction. Cultural elaborations further transformed the voice into a marker of identity. Accent, intonation, and prosody distinguish groups and individuals, functioning as auditory equivalents of facial features. Archaeological evidence of musical instruments, such as bone flutes from the Swabian Jura, dated to over 35,000 years ago, suggests that humans extended vocal capacity into constructed soundscapes, embedding the voice within larger systems of performance. The throat, once reshaped for speech, became the foundation of symbolic culture. Yet questions remain regarding the precise pathways by which these modifications accumulated. Was laryngeal descent driven directly by selection for articulate speech, or was it a byproduct of changes in facial structure and cranial base orientation? Did complex vocal communication emerge gradually from enhanced calls, or did cultural pressures accelerate its refinement once anatomical thresholds were crossed? The evidence points to an interplay of biology and behavior, each amplifying the other in feedback loops of adaptation. The human throat reveals an evolutionary compromise: vulnerability to obstruction offset by the capacity for nuanced sound. The anatomical shift altered not only physiology but also the trajectory of social life, embedding communication at the core of human distinctiveness. Yet while the voice shaped interaction, the choices of individuals, preferences in mates, judgments of beauty, and valuation of particular traits, imposed their own selective pressures, reshaping appearance along dimensions not dictated solely by survival. Natural selection explains traits that directly enhance survival, but many human features cannot be understood without considering the logic of sexual selection. Darwin, in The Descent of Man, 1871, recognized this problem clearly. Certain attributes, from peacock tails to deer antlers, impose metabolic costs and survival risks, yet persist because they increase reproductive success. In humans, the shaping of faces, bodies, and ornaments through mate choice has left visible traces, producing patterns of form and preference that extend beyond mere survival utility. Empirical studies of attractiveness across cultures show remarkable consistency in certain criteria. Symmetry, for example, has long been noted as a key determinant. Randy Thornhill and Stephen Gangestad, through experiments in the 1990s, demonstrated that individuals with more symmetrical facial and bodily features are perceived as more attractive. Symmetry correlates with developmental stability, the ability of an organism to resist perturbations during growth. In this sense, preference for symmetry may function as an indirect signal of genetic robustness. Such judgments occur unconsciously and rapidly, suggesting that they are rooted in evolved perceptual mechanisms. Facial neoteny also plays a significant role. Features associated with youth, large eyes, smooth skin, small chin, are consistently rated as attractive, particularly in women. Anthropologist Douglas Jones's morphometric studies in the 1990s found that composite faces with juvenile proportions were judged most appealing. This preference may reflect reproductive strategy. Youthful features signal extended fertility potential. Conversely, in men, indicators of maturity such as pronounced jawlines and brow ridges can be favored, especially during phases of the menstrual cycle when women are most fertile. This cyclic variation, documented by researchers like Marty Hasselton, illustrates how preferences are sensitive to context, aligning attraction with optimal reproductive outcomes. Body proportions reveal another layer. The waist-to-hip ratio, WHR, in women, quantified by Devindra Singh in 1993, emerged as a reliable predictor of attractiveness across cultures. Ratios around 0.7 are perceived as most appealing, a figure associated with hormonal balance, reproductive capacity, and lower risk of chronic disease. In men, shoulder-to-hip ratio functions similarly, with broader shoulders relative to hips signaling strength and higher testosterone levels. These morphological markers, visible at a glance, became embedded in standards of desirability, channeling selection pressures toward particular body shapes. Sexual selection also influences ornamentation. Secondary sexual traits, such as facial hair in men and breast development in women, extend beyond immediate utility. Human females are unusual among primates in possessing permanently enlarged breasts, not limited to lactation. This feature likely functions as a signal of reproductive maturity and fat reserves, projecting fertility even outside periods of pregnancy and nursing. Male facial hair, by contrast, varies across populations and is subject to cultural modulation, yet consistently emerges as a trait associated with dominance and status. These ornaments, costly in energy and maintenance, persist because they communicate underlying qualities that shape reproductive choice. Voice, too, is subject to sexual selection. Studies by David Putz and colleagues show that deeper male voices correlate with higher testosterone and are rated as more attractive by women, particularly in short-term mating contexts. In women, higher-pitched voices are associated with youth and fertility. Such vocal cues extend the range of attractiveness beyond the visual, integrating multiple sensory channels into mate evaluation. The throat, already adapted for language, became an instrument of sexual signaling as well. Cultural practices amplify these biological signals. Ornamentation through jewelry, cosmetics, or body modification exaggerates natural features associated with attractiveness. The use of red pigments on lips and cheeks, documented archaeologically in Paleolithic ochre deposits, enhances the perception of blood flow and fertility. Neck elongation in certain societies, scarification, and tattooing operate as costly signals, echoing Ahmad Zahavi's handicap principle. Only individuals in good condition can afford the metabolic or social costs of such displays. Human appearance, shaped initially by sexual preference, was further extended by cultural elaboration, embedding choice into systems of meaning. Variation in preferences illustrates the flexibility of sexual selection. While certain traits are universal, others shift with ecological and social conditions. In environments of scarcity, heavier body types may be preferred as indicators of resource access. In contexts of abundance, slender forms may be valorized as markers of discipline or status. Cross-cultural studies, including those by Peter Brown in the 1990s, reveal that ideals of attractiveness fluctuate with subsistence patterns, showing that sexual selection is responsive to ecological variability. The consequences of these choices accumulate over generations. Alleles influencing traits associated with attractiveness increase in frequency, not because they improve survival, but because they enhance reproductive success. Genetic drift, interbreeding between populations, and cultural reinforcement complicate the picture, but the net result is that human appearance bears the stamp of countless acts of selection mediated by desire. Unlike survival traits, which are often constrained by physical necessity, sexual selection produces a diversity of forms, some extravagant, others subtle, but all tied to perception and preference. The human body, therefore, cannot be read solely as a survival machine. It is also a display shaped by competition and choice. Each contour of the face, each proportion of the body, carries potential information about fertility, health, and lineage. Sexual selection ensures that appearance is never neutral, but always charged with meaning, attracting attention and guiding reproduction. Yet preference itself is not static. Shifts in ecology, culture, and technology alter what is considered desirable. The traits once magnified by mate choice are subject to change, their value fluctuating with circumstance. This fluidity leaves open the possibility that features emphasized in one era may recede in another, while new markers of attractiveness may arise, reshaping the visible form of humanity in ways not dictated by survival needs alone. From this interplay between biology and preference emerges a human appearance that is both adaptive and ornamental, functional and symbolic. But the shaping of form does not stop at what the body grows naturally. Humans extend their appearance through deliberate modification, using tools, pigments, and textiles to transform how they are perceived, inscribing culture onto the surface of biology in ways that demand separate consideration. The human body, stripped of fur and exposed to the elements, became not only a physiological surface, but also a canvas for cultural intervention. Unlike other animals, which rely on inherited features alone, humans consistently modify their appearance through clothing, painting, tattooing, and adornment. These modifications do not merely serve protective functions. They communicate identity, status, affiliation, and aesthetic values. The deliberate reshaping of the visible body extends human distinctiveness beyond biology, embedding culture directly into outward form. The origins of clothing are difficult to date archaeologically, as organic fibers rarely preserve, but indirect evidence provides insight. Genetic divergence between head lice, Pediculus humanus capitis, and clothing lice, Pediculus humanus corporis, indicates that clothing was in use by at least 170,000 years ago. This estimate, based on molecular clock analyses published by Ralph Kittler and colleagues in 2003, places the invention of clothing within the period when anatomically modern humans were dispersing into colder climates. Needles made of bone, discovered at sites such as Dzudzwana Cave in Georgia, circa 30,000 years ago, demonstrate sewing technology capable of producing fitted garments. These findings suggest that clothing began as a functional necessity, shielding vulnerable skin from cold and abrasion. Yet clothing quickly transcended function. Ethnographic and historical studies show that textiles carry symbolic meaning, distinguishing rank, profession, and community. Among the Plains tribes of North America, elaborate beadwork on garments communicated tribal identity and personal achievements. In ancient Mesopotamia, textiles of dyed wool indicated wealth and status, with purple, derived from costly murex shells, reserved for elites. The medieval sumptuary laws of Europe codified who could wear specific fabrics or colors, demonstrating how appearance became entangled with political order. Clothing, once an extension of skin, grew into a social language. Decoration of the body itself predates clothing. The use of mineral pigments, particularly red ochre, is among the earliest symbolic practices attributed to humans. At Blombos Cave in South Africa, engraved ochre fragments dating to 75,000 years ago suggest both aesthetic and ritual uses. Experimental archaeology confirms that ochre mixed with animal fat adheres well to skin, producing durable coloration. Such pigmentation may have functioned as sunscreen, insect repellent, or social marker. The deliberate choice to alter the visible body indicates awareness of appearance as a medium of meaning, not merely protection. Tattooing, scarification, and piercing provide further evidence of the body as a cultural surface. The mummified remains of Ötzi the Iceman, discovered in the Ötztal Alps in 1991 and dated to around 3300 BCE, bear carbon tattoos aligned with joints and spinal regions, possibly for therapeutic or symbolic purposes. Polynesian traditions developed elaborate tattoo systems that encoded genealogy and social position. Scarification in West African societies creates raised patterns on the skin, visible at a distance, transforming scars from injuries into deliberate identity markers. These practices impose pain and permanence, underscoring the importance of appearance in human social life. Adornment also extends to the manipulation of natural appendages. Hair, retained on the scalp after the general loss of body fur, became a medium of variation. Styles of cutting, braiding, and coloring have been documented across all known cultures. Beards, plucked or grown, shaped male appearance and carried connotations of wisdom, virility, or disorder depending on context. Fingernails and toenails, though biologically vestigial, are shaped, painted, or elongated in cultural expression, as in the lacquered nails of Ming China or the artificially lengthened nails of aristocratic Europe. The smallest visible structures become canvases for meaning when subjected to human intention. The material culture of decoration reflects technological innovation. Shell beads found at Kafsa Cave in Israel, dating to approximately 92,000 years ago, represent some of the earliest personal ornaments. Perforated animal teeth, feathers, and carved ivory pendants at Upper Paleolithic sites show an increasing complexity in adornment. These objects, carried on the body, altered appearance while signaling group membership. Archaeologist Randall White has argued that such ornaments served as externalized symbols, transmitting social information without words. The body itself became a platform for displaying these objects, turning biology into a foundation for semiotics. The protective and symbolic aspects of clothing often overlap. In Arctic societies, fur garments provide essential insulation, yet decorative embroidery communicates familial ties and aesthetic values. In equatorial regions, where minimal covering suffices for protection, elaborate ornamentation compensates, amplifying symbolic communication. Across climates, the manipulation of appearance reflects not only adaptation to environment, but also the layering of meaning onto the body. Clothing and decoration also mediate the perception of beauty. Cosmetics exaggerate features already favored by sexual selection. Kohl around the eyes, used in ancient Egypt, accentuated contrast between sclera and iris. Lip pigments heightened the redness associated with health and fertility. Powdered skin whiteners in East Asia and Europe emphasized clarity and smoothness, though often at the cost of toxicity from lead compounds. These practices reveal an enduring pattern. Cultural technologies amplify or exaggerate natural signals, reinforcing biological preferences while introducing new standards of attractiveness. The social consequences of modification are profound. Adorned bodies can unite groups or divide them. Military uniforms, from Roman armor to modern fatigues, erase individuality to produce collective identity. Religious dress codes impose visible markers of piety and belonging. Fashion cycles, documented from Renaissance courts to contemporary consumer societies, constantly redefine desirability, demonstrating that appearance is not fixed but mutable under cultural forces. Each change in dress and decoration reconfigures the visual field through which humans recognize one another. Yet the deliberate shaping of appearance does not erase the body beneath. It interacts with it. Every tattoo is placed on a canvas of skin shaped by pigmentation. Every garment drapes over a form sculpted by pelvis, chest, and shoulders. Biological variation provides the foundation upon which cultural invention operates. The visibility of this interaction makes humans unique. Their appearance is at once natural and constructed, inherited and chosen. The layering of culture upon biology raises further questions about how appearance intersects with growth and development. The human body does not present the same surface throughout life. It undergoes dramatic changes from infancy to adulthood. Extended childhood, prolonged adolescence, and the retention of juvenile traits shape how appearance evolves over time, demanding attention to the temporal dimension of human form. One of the most unusual aspects of human biology is the extended period of growth and dependency that characterizes childhood. Compared with other primates, humans develop slowly, remain dependent on caregivers for many years, and only reach full physical maturity after nearly 2 decades. This prolonged developmental window reshapes not only behavior and learning but also visible appearance. The human body presents features of neoteny, the retention of juvenile traits into later stages of life that contribute to its distinctive form. Life history theory provides a framework for understanding this anomaly. Across species, organisms distribute energy among growth, reproduction, and maintenance. Rapid maturation favors early reproduction but limits opportunities for learning and skill acquisition. Slow maturation, by contrast, permits extended development of complex behaviors at the cost of delayed fertility. Humans occupy the extreme end of this spectrum. Gestation lasts 9 months, infancy requires years of dependency, adolescence extends through a decade or more, and lifespan stretches far beyond reproductive years. Each stage leaves visible imprints on the body. The infant head is disproportionately large relative to the body, a result of rapid early brain growth. At birth, the human brain is about 25% of adult volume, compared with 10% in chimpanzees, and it doubles in size within the first year. The cranial vault appears oversized, while limbs are short and trunk rounded. These proportions are not only functional but also visually striking, producing the kinchenchima described by Konrad Lorenz, large head, big eyes, small nose, that elicits caregiving responses. The persistence of such juvenile features beyond infancy contributes to the softer contours of the human face compared with apes. The transition to childhood introduces another anomaly: delayed eruption of teeth. In most primates, permanent teeth appear within a few years, signaling readiness for adult diets. In humans, the mixed dentition stage extends for nearly a decade, with wisdom teeth often erupting only in late adolescence. This prolonged dental immaturity reflects dietary flexibility and reliance on food processing technologies such as cooking, which reduce the need for rapid masticatory competence. The visible dentition, therefore, records the cultural as well as biological rhythms of growth. Body growth follows a distinctive trajectory. Human children grow slowly during early years, conserving energy for brain development, then experience a pronounced growth spurt during adolescence. This spurt, rare among primates, reshapes proportions. Legs lengthen dramatically, shoulders broaden in males, hips widen in females. The timing of these changes is regulated by endocrine systems, with surges in growth hormone, estrogen, and testosterone orchestrating skeletal and muscular development. The visible differences between male and female forms emerge gradually, with the body passing through extended phases of ambiguity. Adolescence also alters skin and hair, visibly marking maturation. Increased sebum production under androgen influence leads to acne, a condition absent in most animals and rare in other primates. Terminal hair growth in axillary and pubic regions creates new visual markers of maturity, while in males, facial hair expands appearance options. These transitions are not merely biological signals, but also deeply embedded in social recognition, as societies across history have marked puberty with rituals, clothing changes, and ornamentation. The appearance of the adolescent body, caught between childhood and adulthood, becomes a focus of cultural definition. The long period of dependency that precedes these changes has profound social implications. Children remain reliant on caregivers for food, protection, and instruction far longer than in other species. This dependency necessitates cooperative care, or alloparenting, involving not only parents but extended kin and community. Sarah H. Ardewey's research on cooperative breeding highlights how the presence of multiple caregivers allowed humans to sustain prolonged childhoods without catastrophic survival costs. The visible presence of young dependents within groups reinforced social bonds, shaping collective identity. Neoteny extends beyond childhood into adulthood. Compared with apes, adult humans retain juvenile cranial proportions, reduced brow ridges, flattened faces, and relatively large eyes. These traits soften appearance, making the adult face more expressive and less intimidating. Stephen Jay Gould emphasized this pattern in his analysis of neoteny, noting that even sexual attractiveness in humans often favors youthful features, further reinforcing the evolutionary persistence of juvenile characteristics. The appearance of the human adult is therefore not merely the product of growth, but of arrested development along ancestral trajectories. The cost of extended growth is vulnerability. Delayed maturity exposes individuals to environmental hazards before they are fully capable of independent survival. Archaeological evidence of child mortality in pre-modern populations is abundant, with high rates of death before age 5. Yet this vulnerability was offset by the advantages of extended learning. The long childhood allowed mastery of language, tool use, social norms, and environmental knowledge. These skills, though invisible in bone, left visible traces in the body's appearance. Faces adapted for speech, hands capable of precision, and postures shaped by cultural practices learned over years. Anthropometric studies illustrate the uniqueness of the human growth curve. In cross-species comparisons, chimpanzees achieve adult size by 12 years, gorillas by 10, while humans often continue growing into the late teens. The visible differences in adolescent limb length, posture, and musculature emphasize how prolonged growth sets humans apart in outward form. This drawn-out transformation produces bodies that carry both echoes of youth and markers of maturity, creating a layered appearance unlike the rapid transitions of other mammals. The extension of childhood and adolescence reshaped not only biology but also aesthetics. Youthful traits became valued across cultures, with cosmetics, hairstyles, and fashion emphasizing smooth skin, round faces, and slender proportions. The cultural magnification of youth reflects deep evolutionary biases rooted in the biology of neoteny. Yet this bias interacts with the undeniable reality of aging, which etches itself onto the body through wrinkles, changes in pigmentation, and shifts in posture. The visibility of age, set against the backdrop of prolonged youth, complicates the human appearance further, introducing a tension between biology, culture, and time. The slow unfolding of growth inscribed the human form with stages that are both functional and symbolic. Each phase alters the face and body in ways that communicate status, maturity, and potential. But the distinctive trajectory of human development also intersected with anatomy in another way. It shaped the hands, organs of manipulation and creation, whose refinement altered not only survival but also the very identity of what it means to look human. The human hand is both an anatomical and symbolic hallmark of the species. Its proportions, musculature, and neural control differentiate it sharply from the grasping extremities of other primates, enabling manipulative precision unmatched elsewhere in the animal kingdom. The hand did not evolve in isolation. Its refinement intersected with upright posture, dietary shifts, tool use, and social communication. The visible distinctiveness of human hands, the opposable thumb, the shortened fingers, the expanded pad surfaces, reflects deep evolutionary pressures that shaped both survival and appearance. Comparative anatomy reveals the peculiarities of human proportions. In chimpanzees and gorillas, fingers are elongated and curved, adapted for suspensory locomotion and knuckle walking. The thumb, though present, is short relative to the palm, limiting its range of opposition. In humans, by contrast, the thumb is long and robust, accounting for nearly 40% of hand length. This configuration enables pad-to-pad precision grip, allowing objects to be held between the tip of the thumb and the tips of the fingers. The anthropologist John Napier, who coined the term precision grip in 1956, emphasized its centrality in distinguishing human hands from those of apes, where grasp remains largely power-based. Fossil evidence pinpoints the antiquity of these features. The hand bones of Australopithecus afarensis, dated to 3.2 million years ago, already show curved fingers consistent with climbing, but with a thumb of relatively human proportions. Later species, such as Homo habilis and Homo erectus, exhibit further refinements. The discovery of a nearly complete hand of Homo naledi in South Africa in 2015 revealed long, curved fingers suitable for climbing, combined with a wrist and thumb aligned with manipulative capability. These mosaics suggest that dexterous hands preceded or coexisted with arboreal adaptations, preparing the lineage for tool use long before locomotor shifts were complete. The internal architecture of the wrist and palm underlies these adaptations. Humans possess a relatively stiff carpometacarpal joint in the second and third digits, stabilizing the hand during precision grips. The trapeziometacarpal joint of the thumb, however, remains highly mobile, permitting a wide range of opposition. Muscles such as the flexor pollicis longus, absent in apes, provide fine control of thumb flexion. These features collectively expand the manipulative repertoire, enabling not only tool construction but also delicate gestures such as threading a needle or writing script. Neurological control mirrors this anatomical sophistication. The sensory cortex of the human brain devotes disproportionate representation to the hands, as demonstrated by Wilder Penfield's cortical homunculus mapping in the mid-20th century. Each fingertip contains thousands of mechanoreceptors, transmitting detailed information about pressure, texture, and vibration. Functional MRI studies confirm that fine motor tasks activate complex cortical networks linking sensory and motor regions. The hand is not merely a physical structure, it is integrated into cognition, bridging perception and action. The archaeological record bears witness to the consequences of this dexterity. The earliest known stone tools, Oldowan artifacts from Gunna, Ethiopia, dated to 2.6 million years ago, display flaking patterns requiring precise grips and controlled strikes. Acheulean hand axes, appearing around 1.7 million years ago, exhibit symmetrical shaping that demanded planning and motor precision. The hand thus became both an agent of creation and a marker of identity, as tools extended the body into the environment. The shaping of stone left visible traces not only in artifacts but in the muscles and bones of the hand itself, which adapted to repeated stresses. Beyond utility, hands serve as instruments of expression. Gestures predate speech in communication, and even today, hand movements accompany verbal exchange across cultures. Adam Kendon's ethnographic research demonstrates that gestures form structured systems, often carrying semantic content independent of words. The visibility of hands during interaction reinforces the communicative role of the body, transforming manipulation into symbol. The palm raised in greeting, the finger pointed in direction, the clasp of agreement. These visible actions mark human social life as much as faces and voices do. Hands also became sites of symbolic modification. The earliest known cave art, hand stencils from El Castillo in Spain dating to over 37,000 years ago, depict human palms outlined in pigment. Such images represent some of the first deliberate portrayals of the body, suggesting recognition of the hand as an emblem of presence. Later traditions continued this motif. Neolithic handprints on pottery, medieval blessings depicted in illuminated manuscripts, and contemporary use of handprints in protest art. The hand, visible and functional, emerged as a signifier of identity and agency. The sexual dimorphism of hands further shaped appearance. Male hands, on average, are larger, with longer ring fingers relative to index fingers, a ratio influenced by prenatal androgen exposure. This 2D, 4D ratio has been linked statistically to various traits, from athletic performance to risk-taking, though findings remain contested. Female hands tend to display smaller dimensions and finer skin texture, contributing to gendered perceptions of delicacy versus strength. These differences, though subtle, contribute to the visual language of human appearance, reinforcing categories of sex and identity. Yet the very refinement of hands carries costs. Injuries to fingers or wrists compromise survival more severely in humans than in apes, whose locomotion remains distributed across limbs. Anthropological analyses of skeletal remains reveal frequent fractures and degenerative changes in hand bones among prehistoric populations, evidence of intensive tool use and manual labor. The specialization of the hand for precision renders it vulnerable to repetitive strain, arthritis, and other pathologies. The same structures that permit mastery also impose fragility. The symbolic weight of hands has persisted across cultural epochs. In religious iconography, divine power is often depicted as a hand extended from the heavens. In law, the handshake seals agreements. In medicine, the healing touch conveys authority. The hand is not only a manipulative organ but also a visible marker of trust, control, and presence. No other part of the body so fully embodies the dual identity of human distinctiveness, practical and symbolic, biological and cultural. The outward distinctiveness of hands, with their expressive gestures and delicate control, integrates seamlessly into the broader constellation of human appearance. Yet this refinement did not occur in isolation. The reshaping of jaws, teeth, and expressions played an equally critical role in distinguishing humanity's outward form, turning even the act of smiling into a uniquely human signal. The human mouth presents a striking departure from the primate norm. Its teeth are small relative to body size, its jaw reduced, and its lips unusually mobile. These structural changes carry both mechanical and communicative significance. They reflect alterations in diet, cranial organization, and social signaling, producing an appearance in which the smile occupies a central role. Unlike the bared teeth displays of other primates, which often function as threats or submissive gestures, the human smile communicates friendliness, reassurance, and affiliation. The mechanics of chewing and the subtleties of expression converge in the mouth, making it one of the most visible sites of human distinctiveness. Dentition provides the clearest evidence of dietary transition. Australopithecines possessed large molars and thick enamel, suited for grinding fibrous plant matter. With the emergence of the genus Homo, teeth began to shrink. Archaeological traces of controlled fire use, including charred bones and heated stones from sites such as Gesher Benot Yaakov in Israel, circa 780,000 years ago, suggest that cooking softened food, reducing the selective pressure for massive jaws. By the time of Homo erectus, molar size had decreased significantly, and with Homo sapiens, the trend reached its extreme: relatively small teeth, lightly built mandibles, and a dental arcade that is parabolic rather than U-shaped. These changes sculpted a flatter face and freed space for the development of the chin, another uniquely human feature. The chin, or mental protuberance, has no clear functional analogue in other primates. Hypotheses for its emergence include mechanical reinforcement of the jaw during speech, redistribution of stresses from reduced dentition, or sexual selection favoring distinctive lower facial contours. Whatever its cause, the chin alters the visual balance of the human face, giving the mouth prominence and lending the smile its characteristic framing. Fossil evidence indicates that Neanderthals lacked a true chin, underscoring its recent evolutionary appearance. The arrangement of teeth also shaped human expression. The reduction of canines eliminated the intimidating tusk-like display typical of many primates. Small, evenly aligned teeth, framed by lips capable of delicate movements, transformed tooth exposure from a threat signal into a sign of approachability. Ethologist Irenos Abel-Ibsfeld documented this contrast extensively, noting that while chimpanzees expose teeth during grimaces of fear, humans expose them primarily in affiliative gestures. The shift in meaning from aggression to friendliness required both anatomical modification and cultural reinforcement. Lips themselves became central to expression. Human lips are thick, fleshy, and mobile, capable of subtle shaping that influences both vocal articulation and facial signaling. The orbicularis oris muscle, encircling the mouth, grants fine control, enabling puckering, tightening, and retraction. This mobility permits not only speech, but also nuanced non-verbal cues. The universal recognition of smiles across cultures, demonstrated by Paul Ekman's cross-cultural studies in the 1970s, testifies to the communicative centrality of the mouth. The combination of reduced teeth and expressive lips made smiles legible and socially indispensable. The cultural elaboration of teeth reflects their symbolic importance. Archaeological finds reveal deliberate modification of dentition. Mayan elites filed and inlaid teeth with jade and turquoise. Japanese traditions included ohaguro, the practice of blackening teeth with iron solutions. Victorian dentistry pursued artificial whitening. These practices highlight how human groups accentuated or transformed the mouth's appearance to align with ideals of beauty, status, or morality. The smile became not just a biological signal, but a site of cultural invention. The vulnerability of human dentition underscores the costs of these changes. Smaller jaws and crowded teeth predispose modern humans to malocclusion, impacted third molars, and dental caries. These pathologies, visible in skeletal remains from agricultural populations, intensified with diets rich in carbohydrates. Yet even in compromised states, teeth retained their social importance. Archaeological evidence of early dentistry, such as drilled molars from Mehrgarh in Pakistan, dating to 9,000 years ago, suggests that maintaining the integrity of the smile carried both functional and symbolic weight. The neurological underpinnings of smiling reveal further distinctiveness. Unlike most facial expressions, which are closely tied to emotional states, the human smile can be voluntary or involuntary. The so-called Duchenne smile, identified by Guillaume Duchenne in the 19th century, involves both the zygomaticus major, raising the corners of the mouth, and the orbicularis oculi, contracting around the eyes. This combination is difficult to fake, making it a reliable signal of genuine emotion. Non-Duchenne smiles, however, can be produced deliberately, expanding the communicative repertoire. The ability to control and modulate smiles underscores their role in negotiation, deception, and affiliation. Cross-cultural evidence affirms the universality of smiling while revealing differences in its social interpretation. In some societies, frequent smiling is associated with politeness and warmth. In others, it may be perceived as insincere or inappropriate in formal contexts. Such variation demonstrates that while the anatomical basis of the smile is universal, its cultural meanings are flexible. The visibility of teeth and lips ensures that the mouth remains a focal point of social interaction, subject to both biological and cultural modulation. The evolutionary trajectory of dentition and expression converged to create a face in which the mouth is more than an organ of ingestion. Its reduced teeth and expressive framing turned it into a communicative tool, transmitting states of mind and intentions. Smiling became one of the most recognizable and contagious signals in human behavior, spreading rapidly through groups and reinforcing social cohesion. Neuroscientific studies using functional imaging show that seeing another person smile activates mirror neurons in the premotor cortex, eliciting unconscious mimicry. The smile, therefore, operates not only as a signal but as a mechanism of synchronization across individuals. The transformation of the mouth and smile carved out a uniquely human dimension of appearance, one that blends utility, vulnerability, and expressiveness. Yet the eyes, positioned just above, carry their own distinctiveness. The unusual visibility of the sclera, the cooperative capacity for gaze following, and the social consequences of eye contact extended human distinctiveness into another domain of expression that reshaped both communication and identity. The human eye is an anomaly within the primate order, not in its optical mechanics but in its outward appearance. While most mammals conceal gaze behind dark sclerae or small irises, humans possess conspicuously white sclera that contrast sharply with pigmented irises and dark pupils. This configuration makes gaze direction highly visible at a distance, permitting precise tracking of where another individual is looking. The visibility of the human gaze has profound implications. It transforms the eyes from a sensory organ into a social instrument, embedding vision within the structure of cooperation. The contrast between human and ape eyes illustrates the peculiarity. In chimpanzees, gorillas, and orangutans, sclerae are pigmented brown or gray, reducing distinction from the iris. Studies by Michael Tomasello and colleagues at the Max Planck Institute for Evolutionary Anthropology in the late 1990s demonstrated that chimpanzees follow head orientation rather than eye direction when interpreting attention. Human infants, by contrast, reliably follow gaze as early as 6 months, using scleral visibility as a cue. This difference is not subtle. It represents a divergence in how social information is encoded in anatomy. The evolution of visible sclera is best understood in the context of cooperative behavior. Humans rely heavily on joint attention, the shared focus of two individuals on an object, as a foundation for communication, teaching, and cultural transmission. The visibility of gaze direction makes such joint attention efficient. Experimental work by Josep Call has shown that when humans and apes are presented with tasks requiring coordination, humans spontaneously use gaze, following to align actions, whereas apes struggle unless additional cues are provided. The human eye, with its conspicuous contrast, thus functions as a cooperative adaptation, facilitating collaboration in foraging, hunting, and social interaction. The morphology of the eye socket reinforces this communicative capacity. The human orbit is relatively wide and forward-facing, increasing the visible field of the eye. Eyebrows, positioned above, frame expressions by exaggerating movements of the upper eyelid, while the surrounding musculature, particularly the orbicularis oculi, permits subtle modulation of eye shape. Emotions such as fear, anger, or joy manifest in part through ocular expression, amplifying the communicative range of the gaze. The combination of visible sclera and mobile surrounding muscles turns the eye into a signal system as much as a perceptual one. Cultural elaboration magnifies this natural distinctiveness. Cosmetics emphasize gaze by darkening lashes, outlining lids, or enlarging apparent iris size with pigments and contact lenses. Historical practices, from kohl in ancient Egypt to modern mascara, exploit the visibility of sclera and iris contrast to heighten expressiveness. In some societies, the deliberate avoidance of eye contact functions as a sign of respect or submission, while in others, direct gaze conveys honesty and confidence. The same anatomy, therefore, acquires different meanings across cultural contexts, but always remains central to social interpretation. The cooperative eye hypothesis, advanced by Hitoshi Kobayashi and Shiro Koshima in 2001, provides an evolutionary framework. They propose that humans evolved depigmented sclera to enhance gaze communication, while other primates retained dark sclera to conceal gaze and reduce competitive exploitation of attention. In highly cooperative groups, transparency of gaze becomes advantageous. In competitive environments, concealment is favored. This theory situates the human gaze within the broader dynamics of social strategy, linking anatomy to patterns of trust and coordination. Neuroscience reveals how deeply gaze is embedded in cognition. Functional imaging studies show that the superior temporal sulcus is specialized for detecting eye direction. The amygdala responds strongly to direct gaze, particularly in contexts of threat or attraction. Mirror neuron systems activate during gaze following, synchronizing perception and action between individuals. The gaze does not merely transmit information, it shapes neural activity in ways that align individuals' attention and emotion. This alignment provides the substrate for teaching, imitation, and cultural learning, all of which depend on shared focus. The symbolic weight of eyes is evident in art and religion. The eye of Horus in ancient Egypt symbolized protection and health. Greek mythology spoke of the all-seeing gaze of the gods. Medieval Christian iconography depicted divine omniscience as an eye within a triangle. Across cultures, the eye functions as a symbol of knowledge, surveillance, and truth. The visibility of gaze direction lends credibility to these associations, for the human eye reveals orientation and intent in ways that few other organs do. Yet the very transparency of gaze introduces vulnerabilities. The inability to conceal attention means that humans are constantly legible to one another. This legibility can be exploited in deception detection, interrogation, or social dominance. Conversely, it can create anxiety, as seen in clinical conditions such as social phobia, where direct gaze is perceived as overwhelming. The evolution of conspicuous eyes therefore imposes costs alongside benefits, demanding cultural norms to regulate eye contact and mitigate tension. Variation across populations highlights both diversity and universality. Eye color, determined by melanin concentration in the iris, ranges from dark brown to light blue and green. These variations arose relatively recently, with genetic analyses tracing the mutation responsible for blue eyes to a single origin in the Black Sea region around 6,000 to 10,000 years ago. While iris color carries no known functional advantage, its visibility against white sclera enhances distinctiveness, contributing to aesthetic preferences and identity markers. The gaze, already a communicative tool, became further diversified by pigmentation, reinforcing its role in appearance. The eye's prominence in human appearance cannot be reduced to optics or survival. It is a social beacon, constantly broadcasting attention and intention, synchronizing individuals within groups. Its visibility alters the dynamics of trust, deception, and attraction, embedding biology within the structure of society. Yet gaze is only one dimension of human diversity. Beneath these shared anatomical traits lies extraordinary variation across populations, differences in stature, facial structure, pigmentation, and build, that render humanity more visibly diverse than almost any other species. If the shared features of humanity distinguish the species from other primates, the variation within the species is no less remarkable. Differences in stature, facial structure, pigmentation, and body proportions are more pronounced than in most large mammals. Whereas many species exhibit relative uniformity across wide ranges, humans present striking diversity, a result of ecological adaptation, genetic mixing, and cultural reinforcement. This diversity contributes to the distinctive visibility of human beings, shaping not only biology but also identity. The distribution of body types across geography illustrates adaptation to climate. 19th-century anthropologists such as Carl Bergman and Joel Asaph Allen articulated what became known as Bergman's and Allen's rules. Larger body mass and shorter appendages are favored in cold environments, while leaner builds and longer limbs prevail in hot climates. Modern population studies support these observations. Inuit populations of the Arctic exhibit compact bodies with reduced surface area to volume ratios, conserving heat. In contrast, Nilotic groups of East Africa, such as the Dinka and Maasai, display tall, slender physiques with elongated limbs that enhance heat dissipation. These morphological adaptations leave visible imprints on populations, inscribing climate into human form. Facial structure also varies with environment. The broad, flattened noses of some populations correspond to humid, hot climates, where wide nostrils aid in cooling inhaled air. Narrow, projecting noses are more common in cold or arid regions, where they function to warm and humidify air before it reaches the lungs. Craniofacial anthropometry, refined through 20th-century measurements and contemporary 3D imaging, reveals consistent correlations between nasal index and climate variables. Such adaptations illustrate how small anatomical changes accumulate into visibly distinctive forms. Genetic diversity underpins these outward differences. Humans share approximately 99.9% of their DNA, yet the remaining 0.1% produces vast phenotypic variation. Single nucleotide polymorphisms, SNPs, influence pigmentation, hair texture, lactose tolerance, and disease resistance. Ancient DNA research has revealed how migrations and interbreeding shaped modern diversity. The sequencing of Neanderthal and Denisovan genomes demonstrated that non-African populations carry between 1% and 4% of Neanderthal DNA, while populations in Melanesia and parts of Southeast Asia carry up to 5% Denisovan DNA. These archaic contributions influence visible traits, including immune response genes and even hair and skin variation. The human body, in its diversity, reflects layers of interbreeding as well as adaptation. Hybrid vigor, or heterosis, further complicates the picture. Populations that intermix often display enhanced resilience and novel combinations of traits. Historical examples include the blending of African, European, and indigenous lineages in the Americas, producing phenotypic variation unprecedented in earlier populations. Such mixing demonstrates that diversity is not static but dynamic, continuously reshaped by movement and contact. The visible spectrum of human difference is therefore a record of both ancient separations and recent convergences. Cultural practices amplify or redirect perceptions of diversity. Standards of beauty vary widely, sometimes emphasizing traits that diverge from local averages. In Japan, historically pale skin was associated with refinement, while in some Pacific Island societies, greater body mass signified wealth and status. These standards shape behavior and appearance through intermarriage patterns, adornment, and even medical interventions. The interaction of biology and culture creates feedback loops. Certain features are preferentially chosen, altering the frequency of traits across generations. Diversity, in this sense, is not merely inherited but also culturally selected. Hair texture and form illustrate this interplay. Straight, wavy, curly, and tightly coiled hair types result from variations in follicle shape and keratin distribution. Environmental adaptation may explain part of this variation. Tightly coiled hair, common in equatorial regions, insulates the scalp from solar radiation, while straight hair, more common in northern latitudes, may conserve heat. Yet cultural valuation of hair types has introduced layers of meaning beyond function. Grooming, styling, and modification, whether through braiding, straightening, or dyeing, transform natural variation into symbols of identity, status, or resistance. The perception of diversity has historically carried political weight. Attempts to classify humanity into rigid races emerged in the 18th and 19th centuries, often tied to colonial expansion. Figures such as Johann Friedrich Blumenbach proposed taxonomies based on skull shape and skin color, laying foundations for pseudoscientific hierarchies. Genetic research in the late 20th and 21st centuries dismantled these frameworks, demonstrating that most genetic variation occurs within populations rather than between them. Visible diversity, though striking, does not correspond to discrete categories, but to continuous gradients shaped by migration and adaptation. Anthropological genetics continues to reveal unexpected patterns. The discovery of high-altitude adaptations in Tibetan populations, involving alleles at the EPAS1 locus inherited from Denisovans, illustrates how localized pressures can produce distinctive physiology and appearance. Similarly, skin fold thickness, body fat distribution, and eye shape exhibit regional variation tied to environment and ancestry. Each adaptation leaves traces in visible form, marking populations with signatures of geography and history. Yet diversity also operates at the level of the individual. Even within a single population, variation in facial features, stature, and body proportions is pronounced. Twin studies reveal heritable components of facial morphology, but also plasticity influenced by environment and nutrition. Malnutrition during growth, for instance, can stunt stature, while abundant nutrition can accelerate it, producing visible shifts across generations. The rapid secular increase in average height in Europe and East Asia over the past century reflects such environmental influence, demonstrating that diversity is both genetic and situational. The extraordinary range of human variation has consequences for perception. Humans are highly attuned to differences in faces, able to distinguish subtle variations in shape and expression. This perceptual acuity underpins both social bonding and group distinction, but also susceptibility to bias and stereotyping. The same neural systems that make diversity visible also shape how it is interpreted, embedding biological variation within social and political frameworks. Diversity ensures that the human form resists uniform description. It is a mosaic of adaptations, admixtures, and cultural overlays, producing bodies that vary in size, shape, and color while retaining shared structures. This visible variability distinguishes humanity not only from its primate relatives, but also from most other large mammals, which exhibit less striking differentiation across populations. Yet the very forces that produced such diversity continue to operate, raising the question of how human appearance might change as environments shift, technologies intervene, and choices alter the trajectory of evolution itself. The human form, the product of millions of years of evolutionary compromise, remains in flux. The visible body is not fixed. It reflects pressures that continue to act: environmental shifts, technological interventions, medical advances, and cultural preferences. If earlier adaptations were responses to climate, diet, and locomotion, the future of human appearance may be shaped by factors unprecedented in evolutionary history. Predicting these trajectories requires caution, yet existing trends reveal directions in which the human body may be compelled to move. Climate change presents one axis of pressure. As global temperatures rise, populations may again experience selection favoring leaner builds and longer limbs for heat dissipation, echoing Allen's rule. Conversely, in regions where cold extremes intensify due to disrupted circulation patterns, compact forms may be advantageous. Skin pigmentation, once closely aligned with latitude, is now complicated by mobility, urban living, and cultural practices. Yet shifts in UV exposure and atmospheric conditions may still influence the distribution of melanin, altering the global mosaic of pigmentation. Such environmental forces, though slower than cultural change, continue to sculpt appearance at the level of physiology. Dietary transformation exerts subtler but significant influence. The global spread of processed foods, combined with sedentary lifestyles, has altered body composition at an unprecedented scale. Rising rates of obesity, metabolic disorders, and dental crowding suggest that modern diets are misaligned with inherited physiology. Over generations, artificial selection through medical technology may prevent natural filtering of such traits, allowing malocclusion, reduced jaw size, or predisposition to metabolic dysfunction to persist. Orthodontics and surgery already reshape faces, blurring the boundary between natural and engineered appearance. The mouth, once reshaped by cooking, may again be altered, this time by technology rather than fire. Medicine and biotechnology introduce forces absent from earlier evolution. Vaccination, antibiotics, and advanced healthcare reduce mortality from diseases that once exerted selective pressure. Genes that previously limited survival may now persist, altering the genetic landscape. At the same time, reproductive technologies, egg freezing, in vitro fertilization, and potential germline editing permit deliberate choice over inherited traits. The CRISPR-Cas9 system, first applied to human embryos in 2015, raises the possibility of directed modification of appearance, from pigmentation to stature. While ethical constraints limit current applications, the capacity exists for future generations to inherit bodies shaped as much by decision as by adaptation. Cultural selection operates alongside these technologies. Standards of beauty, mediated by global media and digital platforms, influence partner choice across continents. Preferences for body shape, skin tone, or facial symmetry may narrow diversity, amplifying certain traits while suppressing others. Cosmetic surgery, already widespread, accelerates this process. Procedures altering eyelid folds, jawlines, or nose shape demonstrate how culture can impose rapid, visible change, compressing what evolution would stretch across millennia into a single lifetime. The human form becomes both inherited and manufactured, biological and designed. The digital environment introduces another dimension. Increasing immersion in screens and virtual interfaces may alter the body indirectly. Ophthalmologists document rising rates of myopia worldwide, particularly in East Asia, attributed to reduced outdoor light exposure and prolonged near work. If such conditions persist, future populations may exhibit changes in eye shape or reliance on corrective technologies. Prolonged seated postures, reduced physical activity, and dependence on artificial lighting reshape musculature, bone density, and circadian rhythms. These modifications, though driven by lifestyle rather than selection, accumulate into visible differences over generations. Space exploration extends the horizon of change. Extended habitation in microgravity leads to bone demineralization, muscle atrophy, and redistribution of fluids, visibly altering posture and facial appearance. NASA's twin study, conducted on Scott and Mark Kelly between 2015 and 2016, documented measurable physiological changes after a year in orbit, including shifts in gene expression and carotid artery thickening. If humans colonize extraterrestrial environments with reduced gravity, body proportions may elongate, and musculature may adjust to altered loads. Skin tone may change under different solar spectra, and protective technologies may exaggerate or mask natural variation. In such contexts, human appearance may diverge across planetary environments, producing lineages visibly distinct from one another. The prospect of genetic convergence complicates this divergence. Global migration and intermarriage blur traditional population boundaries, producing novel combinations of features. Anthropologist Stanley Garn predicted in the mid-20th century that increased mixing would lead to greater homogeneity of human appearance. Contemporary genetic analyses confirm extensive admixture, particularly in cosmopolitan regions. Yet the direction of this convergence remains unpredictable, shaped by patterns of movement, preference, and reproduction. The visible face of humanity may both diversify and homogenize, depending on context. Artificial augmentation extends beyond medicine into enhancement. Prosthetics, implants, and wearable devices increasingly integrate with the body. Cochlear implants, retinal prostheses, and robotic limbs already alter visible form. Future technologies may render the line between biological and synthetic indistinct, with appearance shaped by choices of integration. Cyborg traits, skin-embedded sensors, cranial interfaces, exoskeletal supports, may become part of visible identity, reshaping the very definition of human form. Such modifications raise the possibility that diversity of appearance will not only reflect genetics and environment, but also technological adoption. The trajectory of human appearance is therefore not linear but contingent, shaped by forces natural, cultural, and technological. Each factor imposes pressures that may reinforce or counteract one another, producing outcomes that cannot be wholly anticipated. The visible form of humanity will continue to change, whether subtly through shifts in diet and climate or dramatically through engineering and augmentation. What remains constant is the body's role as both biological substrate and social surface, the site where adaptation, identity, and invention intersect. The question that persists is not whether humans will look different in millennia to come, but in what ways the pressures of ecology, culture, and technology will intersect to sculpt that difference. The face, the skin, the posture, the gaze, all are open to alteration, carrying forward the unresolved experiment of human appearance into conditions no previous generation has encountered.